Okay, we're ready to now bring this sort of all together. This is where we sort of take everything we've learned this lecture, and as we move into genetics, start to put it all together. Hopefully, that's our goal here. We can now put together some things that we kind of know that we've learned in this class, and that we kind of brought in knowing before we even came to this class. So now we've talked about DNA, and you know about the structure of DNA with the double helix molecule. James Watson, Francis Crick, Maurice Wilkins, Rosalind Franklin, really who should be given credit, although she wasn't included in the Nobel Prize because unfortunately she passed away before it was awarded. All had tremendous contributions to discovering the structure of DNA. So here's my DNA, and one of the things about DNA that that we haven't really talked about yet is that when you first see a cell, if you look through a microscope and you look at a cell, you probably could see the nucleus, but that's about all you can see. It's really hard. You can't really see chromosomes in a normal cell unless it's going through division. So normally what you see is you'll see a whole bunch of strands, and it'll look sort of grainy in there, and that's because what happens is my DNA is in there, and it's wrapped around these proteins, and the proteins are called histones. So the DNA is sort of contained and wrapped around these proteins, kind of like yarn almost, and then the histones and the DNA, those are coiled up then into this sort of strand material that you can see here, and that's the chromatin. So normally what you see in a cell during its normal cycle is you'll actually see the chromatin. You won't actually see chromosomes unless it goes through division. And then when you first see them, actually what you'll see is a duplicated chromosome. So we'll talk about that coming up. So once again, here's my DNA, here's my histones, here's my chromatin. And what will happen is this will duplicate. It'll go through DNA replication, which we mentioned before, using DNA polymerase and DNA ligase to copy all the DNA. And when you copy the DNA, it will then condense after that. And so the first time you actually see chromosomes, what you see is really it's doubled at that point. And you have two sides to it, and the two sides are what we call chromatids. And those are copies of one another, and they're connected at some point, sometimes in the middle or sometimes on the end, and that is called a centromere. And then the two chromatids are what we call sister chromatids. Okay, so we'll talk about that as we move along as well. The other thing we talk about is this thing called a karyotype. So a karyotype is a picture of your chromosomes. So if you look here, when a cell condenses and forms its chromosomes as it goes through division, one of the things you can do is you can actually get a picture of these, and you can look at the chromosomes, and you can stain them, and they have different colors. And in this particular case, this is a human karyotype, and so it has 23 pairs of chromosomes. It has what they call autosomes, which are, say, chromosomes 1 and 2, which are homologous. And homologous chromosomes are those that carry the same type of genetic information in one location relative to another. So, for example, you have two chromosome 1s. The information on that part of chromosome 1 should be kind of the same as the information there. So, for example, if that information codes for eye color, on that chromosome, this one codes for eye color as well. They might be different. One might be brown eyes and one might be blue eyes, for example, or they might be even the same. But certainly, as long as your parents are not the same person, if your mom and dad are the exact same person, then your chromosome 1 and chromosome 1 on homologous pairs will be exactly the same, but it shouldn't be. So what will happen is we have our two chromosome 1s. Those are called autosomes. Then we also have our sex chromosomes, and those are not homologous unless you're a female. So you notice the X chromosome and then the Y is actually very different. So the Y is a little tiny chromosome there, and then there's an X. So if you're a male, you have one X chromosome and one Y, and if you're a female, you have two X's. That's true in humans, but that can vary depending on other animals where the chromosomes aren't the only effect that are involved. We won't go into that in this class, but that's basically true for other animals. Could be. Okay, so chromosome 1, for example, if you've got eye color information there and eye color information there, chromosome 2 means at that same location on it, there's something totally different. This might be related to your height, for example. Okay, and the Human Genome Project has allowed us to map out kind of what each part of each chromosome is responsible for doing. Okay, and then where they're connected is what we call the centromere. So you can tell, for example, in a karyotype, if a person has an extra chromosome 21, for example, that would result in what's called Down syndrome. So there are certain genetic conditions we'll talk about where you can actually tell a person's going to have a genetic issue of one sort or another because of the number of chromosomes or a part of a chromosome missing. Most of the genetic issues we'll talk about, though, are in the code. So what you can't see here is the actual sequence or the code. To do that, we need to use other techniques we'll get at later on. Okay? Now, what we're going to do is we're going to take this cell. So we have our one cell here. And what we're going to do in this case is we're going to take our, I guess we should start over here. We're going to take our one cell here. And we're going to take one cell and we're going to copy everything in that cell and end up getting two cells out of that. That process is called mitosis. So mitosis basically allows me to take one cell, copy everything in it, divide it up, and get two cells. And this is used primarily for growth. So you start off when you're born, actually before you're born, when you're conceived, I should say, you start off as one cell. And then that one cell becomes two. And then those two cells become four. And that continues on until you become a little person and then you're born. Okay? It's a little more complex than that, but that's a good start to it. So what we're going to do is we're going to take that one cell, and the cell cycle goes through these three phases here. One is G1, then S phase, and then G2. G1 stands for either, you see both these terms, either growth or gap, 1 and 2. And during G1, your organelles are being copied. Your organelles like your ribosomes, your mitochondria, your endoplasmic reticulum, and all the organelles get copied during G1, and that continues through S phase and in through G2. During S phase, in addition to those processes continuing on, DNA synthesis occurs. So that's where we copy the DNA. So S phase is where the DNA gets copied. Okay. Now, I should also point out that we have two sort of ways we can define cells based on the number of chromosomes they have. We have diploid cells and we have haploid cells. Haploid cells have one set of chromosomes in them. Okay, and we usually use a letter N to represent that. And diploid cells have two sets of chromosomes, so we do 2 times N. Okay, and N therefore stands for how many chromosomes are in a set. In the case of a human has 23 chromosomes in a set, for example. So a haploid cell, like a sperm or egg, would have 23, but a diploid cell, like on your skin or in your liver, would have 46. So most of your cells are going to have 46 chromosomes in them, but the number of chromosomes varies depending on what kind of organism you have. So you might have a horse that has 50 chromosomes or a fly that has 4 chromosomes. The number of chromosomes differs depending on the species. Okay? So what we're going to do is we've copied the DNA. During S phase, we copy the DNA. During G1 and S phase and G2, we copy the ribosomes and so forth. And now what we're going to do is that all makes up what we call interphase. So during interphase, what we start to see is we see a cell with my DNA that at this point has been copied. It's in the form of chromatin. Remember, that's DNA wrapped around the proteins. And then we'll see these centrosomes here with these centriole pairs. These are specialized proteins that are going to divide and they're going to travel from one side to another. And so what you see, that's interphase. As we go into prophase, this chromatin will condense into chromosomes. So now I can see my actual chromosomes, and once again these are duplicated at this point. So here's my centrosomes here, okay, and there's a centrosome and a centrosome, and we can see these proteins. These proteins are called microtubules. We can see these proteins starting to reach across the cell. My nucleus is starting to dissolve so that the chromosomes can be sort of in the middle of the cell like that. And so that all happens during prophase. My chromosomes condense into chromosomes. My meiotic spindle fibers start to form. Those are the proteins stretching across the cell. My nucleus dissolves, and that's what I see at prophase. During metaphase, what we'll find is all of our chromosomes will line up in the middle. Okay, so metaphase, the chromosomes line up in the middle at what they call the metaphase plate or line. So you see both those terms, metaphase plate or metaphase line, and they line up like that. And then what's going to happen is during anaphase, my sister chromatids are going to separate. So sister chromatids separate chromatids. We're going to separate. And when they separate, those will then become daughter chromosomes. So what will happen then is as they separate, I'll end up forming during telophase and cytokinesis. This cell will then divide into two cells. A nucleus will form around this set. A nucleus will form around that set. And what I end up with is I took my duplicated chromosomes, and each one now has one of those, one of those, one of those, and one of those. Say 2 chromosome 1s, 2 chromosome 2s in each cell. And so when I'm done, I end up with 2 diploid cells. I started with a diploid cell, copied everything in it, and now I have a diploid cell at the end. Okay, so the main goal, what we do in mitosis, this can actually be slightly different depending on what kind of cell you have. So fungi and things like that actually have a couple of exceptions to this. But for the most part, generally true in humans, for example, what we're trying to do in mitosis is we start off with a diploid cell, we copy everything in it, and we make 2 diploid cells. And you use this type of reproduction of cells for growth, to make new cells, to make new skin cells, for example. Okay, so that is mitosis. Now, the other cell division we can talk about is called meiosis. So what happens in meiosis, we're going to take a diploid cell and we're going to copy everything in it, but we're going to divide it up, and our goal is to make haploid cells. Haploid cells have 1 set of chromosomes in it. So if you take, for example, a male and you take a female, so your mom and dad, for example, if you will, I know a disturbing thought maybe, but you have the sperm and you have the egg. Well, the sperm has 1 set of chromosomes in it. The egg has 1 set of chromosomes in it, 23 and 23. If you don't go through meiosis and you go through mitosis, you have 46 and you have 46, which would get together and you would have 92 chromosomes in your cell. And you might think more is better, but it turns out that is not true. Having too much DNA really, depending on the species, is very true in humans, really wreaks havoc on the development of a growing baby. So having a whole extra set of chromosomes, 2 sets, is really really profound. Even having just 1 extra chromosome has pretty large effects. So what we're going to do is use another process where we take the DNA, we're going to copy everything in it, but we're going to divide it twice, and that's called meiosis. So what we're going to do, we do the same thing. We go through, first of all, G1, we go through S, and we go through G2. So that part looks the same. So we end up during interphase having, once again, our centrioles are forming. We have our chromatin. DNA is getting copied. During prophase 1, kind of looks the same. You have your chromosomes condensing where you can see them. You have your spindle fibers forming. But something different happens here. In prophase, the homologous chromosomes are going to pair up. Okay? They're going to pair up with each other. And when they pair up with each other, they're going to form these structures called a tetrad. So these are your 2 chromosome 2s, for example. They're going to form these tetrads. And what's interesting about this is they're actually going to swap parts. They're going to switch pieces with one another. I'll show you that in a second. Okay? So they're going to switch pieces with each other. And that's going to give you then a chromosome 1 or 2 or 3 that is not like your mom's or your dad's. It's going to be a mixture of your mom's 2 and your dad's 2. Okay? So this is called recombination. It gives you genetic variation. So you don't get a chromosome 1 that's exactly like your dad's chromosome 1, for example. You get a chromosome 1 from your dad that's a mixture of his 2 chromosome 1s. Okay? So that's in prophase. And that event, very important event, is called crossing over. Okay? Crossing over. Don't need that in there. During metaphase 1, this is also different. The homologous chromosomes are going to line up in pairs. So my 2 chromosome 1s are going to line up together, my 2 chromosome 2s are going to line up, and my 2 chromosome 3s. So that during anaphase 1, instead of the sister chromatids separating, they're not going to separate. Instead, I'm going to pull the whole pair over that way. So I'm going to take a chromosome 1 that's duplicated, another chromosome 2, and a chromosome 3 that are all duplicated during anaphase 1. Let me show you this really quick. This is the crossing over I mentioned. So here's chromosome 1 that, say, your dad has, and this is chromosome 1 that your dad has also, that he got from his grandparents, okay? Or from his parents, I should say, your grandparents. When they go through meiosis, these get together like this, and they swap pieces. So you see, they've swapped parts. Because what's going to happen is, you're only going to get one of these pieces. You're not going to get the whole one because you get your other set from your mom. All right? So what happens then is during telophase 1 in cytokinesis, those then divide. And I end up with, at this point, when these divide, I end up with, at this point, 2 haploid cells. So these cells are haploid, but they have duplicated and connected sister chromatids. Okay, so the sister chromatids remain attached. I have 1 duplicated chromosome 1, 1 duplicated chromosome 2, and 1 duplicated chromosome 3 in each cell. Okay, so they're haploid. Then I'm going to go through all of these steps again. And these steps are essentially like mitosis. So what I'm going to do during prophase 2, metaphase 2, anaphase 2, and telophase 2 and cytokinesis 2, I don't go through the cell cycle again. I don't go through interphase again. I go right into prophase. I take that cell there and I start forming my meiotic spindle again. The chromosomes all line up in the middle, but they're haploid this time, so I only have 1 of each instead of 2 of each. And you notice the crossovers occurred, so they've swapped parts. During anaphase 2, the sister chromatids separate. And then I form a new cell around that, and what I end up with 4 haploid daughter cells. Each one is in value. And if this was a human, it would have 23 chromosomes in each one. Okay, so just to highlight again the points that are quite different in mitosis and meiosis. First of all, in meiosis, during prophase 1, I have this crossing over event where the tetrads get together and I swap parts. That does not happen in mitosis. Okay, so in mitosis, the sister chromatids don't really swap parts. That is a very common event in prophase 1 of meiosis. Okay, during metaphase of mitosis, all the chromosomes line up in the middle, and during anaphase, those separate, and I end up with 2 diploid cells, 2N. Okay, during metaphase 1, the chromosomes line up in pairs, the tetrads line up, and instead of them splitting sister chromatids, the whole chromosome goes over, and I end up with 2 haploid cells, but they're duplicated, so that goes through the process again, and when I'm done, I end up with 4 haploid cells. So in this case, I can end up with, let's say, a sperm and an egg, and when those get together, you end up with 2 diploid cells. Okay? Last slide I'll show you here, just to show you the difference here. These are sperm here, so you notice the size here of the sperm, and this is the egg of a human. Big giant egg, little tiny sperm. Males tend to make fairly small sperm with almost nothing in them except the genetic information in them, and then the egg has pretty much all the nutrients in them, all the mitochondria and all that, and so the egg is a much bigger cell. Okay, so again, those 2 processes are mitosis, which is used for growth, and meiosis, which is used to make sex cells or gametes.